Hi, my name is Abby, and in today's video, we'll be looking at developmental networks. Developmental networks are pathways of genes and regulators that determine if a certain phenotype will be expressed. We'll be looking at this developmental network that regulates the formation of spikes on certain beetle species. The S in our pathway serves to activate the transcription of gene P in our chain. P, in turn, activates the production of gene I. If I is successfully activated by P, then it will act as an inhibitor and block the transcription of K. K is an inhibitor of gene E. If K is present, transcription of E will not take place. If K was inhibited by I in an earlier step, then transcription of E will occur. Assuming our pathway ends up with the production of E, the cell will activate the formation of spikes in the ectoderm. If no E is produced, there will be no spikes. If there is a mutation in our network that causes P to be non-functional, then transcription of I will not be activated. Since there is no I to repress the creation of K, K will block the transcription of E, and the cell will not form spikes. A mutation that causes the I of our chain to be faulty would also lead to a spikeless beetle. This would be because, like the previous mutation, nothing would be present to block the formation of K, which would inhibit the transcription of gene E. Without E, there would be no formation of spikes. A mutation in both I and E would also lead to no spikes. A faulty I, as we saw previously, would lead to no spikes on its own due to the formation of the inhibitor K. Since E is also faulty and further downstream in the network, it would not be able to produce the needed product to facilitate the formation of spikes anyway. The last mutation we will look at is if both S and K were not functional. In this situation, we can look at what K would do as it is further downstream in the network than S. The functionality of S would not matter as it wouldn't change the outcome because although gene P would not be activated and thus I would not inhibit K, gene K would still be not able to function. With K being mutated, it is unable to inhibit the transcription of E, allowing for the formation of spikes. We know that not every cell on the beetle's ectoderm will have spikes. This is partly because some of the ectoderm cells, like nerve cells, are internal and would have epigenetic tags on their DNA, turning off the network so no spikes form inside the body. Another factor is variable expressivity. We could hypothesize that in some of the ectoderm cells, random mutations or epigenetic tags turn off one or more of the genes in the developmental pathway, making some cells form spikes and others spike less, varying the expressivity of our phenotype.